Okay, so let's go ahead and start talking about some of these different groups of protostomes. Definitely make sure you have gone through the protostome basic lecture, kind of the key features that are protostomes before you guys jump into these. That lecture there is meant to give you kind of that roadmap about the protostomes and how things are organized underneath them. So when we get into these different phylum, I want to just jump into the phylum and talk more about the phylum than all those key things. Okay, so when we look at this protostomes, sometimes you will see the term bilatera thrown out there associated with certain animal groups. Bilatera means the organisms have bilateral symmetry. So you cut it through the center in all different directions and you have right and left sides. That's the idea of bilateral symmetry. So again, just wanting to introduce that in case you see it in the literature or your book or reference somewhere. But again, protostomes. I'm not going over this. Fill it out. What is the key feature to identify something as a protostome? You guys should have this in your memory banks. If not, get it in there. What are the two main groups of protostomes out there? Again, go back to that the referencing lecture about protostomes. Two main groups there, Sporilia. Within Sporilia, we have Platyzoa, Lophocotrozoa, and then the other key group, kind of parallel to Sporilia, will be the Ectozoa. What is the identifying feature of Ectozoa? So now that we can finally get down to the phylum level, let's talk about the Platyhelminthes. So Platyhelminthes are part of Sporilia, Platyzoa, and then we put them into phylum Platyhelminthes. These are the flatworms. These are animals that have one of the most complex life cycles among the animal world. So they often have multiple hosts that they use throughout their life cycle. Let me move this. So then there's going to be a bunch of other key features, characteristics I want you guys comfortable with in regards to the flatworms. So we'll start with flattened bodies. It kind of goes without saying, hence why they're called the flatworms. They have an incomplete digestive system. Okay, so they do obviously use nutrients, but they don't have this full digestive system. They do exhibit, by and large, make sure I spell this right, a thing called cephalization. Cephalization means you have a head or a head end, something that defines the front and the back end of the animal. The head end will often have a simple brain within it and sensory organs. Not to the complexity of what we see in higher animals, but things that help them detect stimulus, eye spots, and things that can detect tactile, mechanical, light levels, etc. tend to be concentrated in this head region. When we look at the body plan, it's made up of 3 tissue layers. So we talked about that. We talked about that with development. When they get their oxygen and use or get rid of carbon dioxide, gas exchange is primarily through diffusion. So oxygen diffuses through the body wall, carbon dioxide diffuses out. And by and large, a lot of them are hermaphroditic. So there's a mutual exchange of sperm. Some of them can go asexual and reproduce through fragmentation. Okay, so kind of big general features for the members of phylum Platyhelminthes. And then what we're going to do now is just talk about some different examples of members of phylum Platyhelminthes. There's going to be some different hyperlinks in here. Check it out. Some little videos I found. Again, if the videos aren't working, please let me know so I can find a new one. You never know, video works today and then the link gets pulled tomorrow. So hard to know. I hope they will all be working by the time you guys are watching these things. So some entertaining videos. So, all right, so let's take a look at the first group of platyhelminths. These guys are the Turbellaria. Now, Turbellaria are free-living members of the platyhelminths. A common one is known as the Planaria. Some of you guys, depending on high school biology classes, you may actually have worked with these things. So basic body plan, it's flattened up to the top here. You can see these little eye spots. These are not true eyes. They just detect light levels. The weird thing is the mouth is on the underneath side of the body. So they crawl over the top of food. The pharynx and the mouth protrude down below. They suck up food, goes into a very simple digestive system, break down the nutrients. Diffusion of the nutrients causes it to absorb throughout the body. Waste gets actually diffused out these tiny little, there's no anus, but out these little tubes on the side of the body here. They're known as flame bulbs that help distribute and excrete the waste product. They do have a nervous system. They have a, we could kind of call it a brain. They can learn. So if you ever raise planaria, you can teach it to find food, to work through mazes, things like that. As we mentioned, they're hermaphroditic. They have an ovary or ovaries and testes. They do not want to self-fertilize. They want to trade sperm with another. So it's a huge advantage if you're a hermaphrodite. You can mate with anybody in your population and increase your reproductive potential. Okay, so kind of some neat things. One really cool thing, if you ever have the opportunity to work with them, you can take a flatworm and you can slice its head in half this way, and it will actually grow a new half of its head over here like this on one body. And then you want to get crazier, slice it again, and you can try to grow a four-headed planaria. So some kind of fun stuff you can do with these animals in the lab. So a lot we can learn from them as well. So, all right, another group within the platyhelminths are what we call the trematodes. Now, trematodes are parasitic flukes. Right? These guys are not our friend. We tend not to like the trematodes. Turbellaria, no big deal. We have no concerns about those guys. But parasitic flukes, yeah, be a bit problematic. Oops, this. So the problem with them is if we get an infection, a trematode infection, it's tough to deal with because their epidermis, their skin, is actually resistant to the digestive enzymes of the host. They use suckers and hooks to hang on the intestinal wall or the internal membranes of the host. So it's hard to get rid of these suckers if they're attached and they're living inside of you. So an example of a nasty trematode is this thing called the Oriental liver fluke. Now, this is one of those things that has two different hosts. That's why we talked about the life cycle being complex. So let's start with the little larva. Excuse me, ciliated larva swimming around in an aquatic, wet, moist environment. It needs to get into a snail, specific species of snail, and use that snail as a host to grow and mature into the motile larva. Now, once it's become the motile larva, it then looks to infect and parasitize humans. So they'll swim around, they'll burrow into your skin, through your skin, burrow into your skin layers, and then they get into the vascular system. And what they're going to do is they're going to burrow their way into the gut. So basically, they're going to try to get their way into your digestive system. Male and female will reproduce. They'll drop their eggs in your intestinal tract. you'll poop out the eggs. The eggs go into the aquatic system, and the life cycle, well, fertilized eggs, the life cycle continues again. So not a friendly thing. So the goal is to try to figure out, how do we break this life cycle? Can we get rid of them in a human host? Sure. Can we get rid of them in the environment? A little more difficult. So are there strategies where we can get rid of these things and decrease parasitism on humans? A person gets infected without medical treatment, these liver flukes can live 15 to 30 years inside the host, just constantly parasitizing the host and leading to some long-term health issues. All right, so the one we're probably more familiar with and more likely to come across will be Saytoto. Now, these guys are the tapeworms. So Saytoto are going to be vertebrate parasites. So we see these things parasitizing vertebrates, animals with backbones. Okay, so all sorts of interesting features and characteristics that they use to make them successful. Huge. Biotic potential. When we look at eggs, an adult can produce about 600 million eggs in a given year. That is massive, massive biotic potential. The problem we'll see is they absorb nutrients from the host. So they parasitize us, they get inside the intestinal tract, and then as you consume nutrients, they just absorb it. It diffuses right through their body wall into the parasite, gives them energy. So often you make the joke, wow, you're eating all the time, yet you're losing weight. Do you have a tapeworm? Stupid as it is, female models years ago used to take tapeworm, we call them proglottids, these little sections. They would take these sections, consume them, allow the tapeworms to develop inside of them so then they could eat and not gain weight. It was stupid, crazy, crazy stupid that people would do this because, yeah, okay, I can lose a lot of weight this way, but then how do you get rid of the tapeworm? That's the problem. Once it's in you, it doesn't want to go away. So it uses these hooks and suckers to actually hang on to the intestinal wall of the host. And then the body stretches out and they have all these little segments. The segments are what we call proglottids. Now, the bigger the proglottid, the further away from the head it's going to be, or what we call the scolex. So these really big ones down here are what are referred to as gravid proglottids. Gravid means it's full of eggs. And the further they are away from the host, the head, the scolex, where it attaches to the body wall, the bigger they get, the more eggs they have. So the plan here, the goal is, as these tapeworms elongate, and they can get up to 2,000 segments, 10 meters. It's about 33 feet in length inside your intestinal tract. The further they are from the attachment point, the more likely they are to break off. So that's a strategy. A couple proglottids break off, they come out in the feces of the host, and then they get picked up by another host. Life cycle continues. Okay, so here's how the life cycle works. So let's just say proglottids are in the environment and they're sitting here on the grass. The zygotes are sitting on the grass. Cow comes along, chews on the grass, now is infected. Those zygotes develop, they start building up inside the cow. They can then encyst into the muscle tissue of the cow and actually burrow into the tissue of the cow and just remain dormant. We then eat that cow, went out, had steak, hamburger, etc., and you're potentially eating those proglottids. So in the part 2 of the lecture, I'm going to talk about the concerns about eating beef if it's not cooked well enough.